right. It is a few minutes after the hour, so I'm going to get started. Good morning, good afternoon, everybody. I guess maybe it's not afternoon this time, but thank you for joining us for this HDSA research webinar. I am Dr. Leora Fox. I'm the Assistant Director of Research and Patient Engagement at HDSA. I am joined also by our Chief Mission Officer, Dr. Eric Johnson. And today we're going to be hearing from Uniqure about their ongoing GeneTRX studies of AMT-130. This is the first experimental gene therapy for Huntington's disease. I'll note that in December of 2023, they presented their latest data to an investor audience, and we're very happy to be able to provide a platform for them to present to the wider community and to give you all an opportunity to learn more from them directly and to ask questions. So I'll introduce our speakers in just a moment, but first I just want to remind you about how we go about our Q&A at HDSA webinars. So first, our panelists are going to be making a presentation. They'll address questions after they present their slides, but you can type your questions into the chat at any time. It's the chat box in the Zoom toolbar. This is a closed chat, so only HDSA and our panelists are going to be able to see what you have asked. We will then pose the questions to the panelists at the end. We do try to focus on questions with broad relevance and to keep things anonymous rather than personal. There's a lot of attendees today, so if we aren't able to answer all of your questions, you're welcome to reach out to HDSA, and we're happy to help follow up and get our speakers involved. So we are recording today, so if you want to watch this presentation later on, or if you want to send it to someone else, you can go to hdsa.org/researchwebinars, or you can go to HDSA's YouTube channel using the button at the top of our homepage. Usually that's up in a day or two. To learn about other upcoming HDSA webinars, you can join our mailing list. You can check out hdsa.org/researchwebinars. If you're interested in other types of information about Huntington's disease or other programs and services that we offer, you can see that at hdsa.org/support. If you want to help support the programs that we offer to those affected by HD, you can visit hdsa.org/donate. And for specific research info and opportunities, you can follow HDSA's social media, our research blog. You can also go to HDSA's hdtrialfinder.org to learn more about currently recruiting clinical trials, as well as our surveys page, hdsa.org/surveys. There's lots of different ways to contribute to HD research from home. So now I'm very pleased to introduce our speakers. Dr. Walid Abi-Saab is Unicure's Chief Medical Officer, and he joined the company in 2023, but he has more than 2 decades of experience in global drug development, including serving as Chief Medical Officer at Galapagos and holding various leadership positions at other pharma companies. And under his leadership, more than 30 molecules have advanced through clinical development, leading to several approvals in the US, EU, and Canada. And having met Walid, I can say that he's certainly approached this role with great thoughtfulness and compassion for the HD community, so I'm glad to be able to introduce him to you today. As for Dr. Ed Wild, I won't tell you that he needs no introduction, but many of you may recognize him. Ed's a neurologist and researcher at University College London who wears many hats in the HD community since 2005, including leading a research team focused on clinical trials of new treatments, studying cerebrospinal fluid to understand HD, and co-founding HDBuzz.net, which is a leading source of HD research news in understandable language. He is a lead investigator in Unicure's HD-GeneTRX trials, which is why we have the pleasure of seeing him today. So welcome to Walid and Ed, and thank you both so much for being here. And I'll let you share your slides.
Thank you, Leora. Our slides on? Dan is going to help me with that. Thank you, Dan. So here we go. All right, very good. So, good morning, good afternoon, everyone. My name is Walid Abi-Saab, and as Leora said, I'm the Chief Medical Officer at Uniqure. I've been only here for a little over 6 months. I have to say I'm thrilled to be with you today to present an overview and an update on our clinical trials with AMT-130. Next slide, please. So this is our disclaimer slide. What you should know is that I am an employee of Uniqure. that I am going to be talking about a study about a program that is in development that is not necessarily have demonstrated full efficacy. And if you have any questions about this, you should be talking to your physician first before you make any decisions about that. Next slide. So before I start my presentation, I really would like to give a heartfelt thanks to all the people with Huntington's disease who took part in our trials, to their families, their caregivers, their doctors, and honestly, the larger Huntington disease community. Without you, none of this would have been possible. So thank you, really. Next slide. So who is Uniqure? Well, we are a company whose mission is to develop gene therapies for severe genetic diseases. And in doing so, we aim to transform people's lives. Next slide. At Uniqure, we consider ourselves pioneers. We have been blazing new paths in gene therapy since we were founded more than 25 years ago. In 2012, we got approval for the first gene therapy ever in Europe, a product called Glybera. In November of 2022, the gene therapy we developed for hemophilia B, the first of its kind, was approved in the US and in Europe. But most importantly for everyone here, in 2020, we initiated the first gene therapy clinical trial ever for Huntington disease. Uniqure operates out of 3 locations: Amsterdam, Lexington, Massachusetts, and Basel, Switzerland, where I am based. Next slide. So what exactly is gene therapy? To put it very simply, gene therapy is the method of using genetic material to treat or prevent disease. Gene therapies have been in development for decades, and over the past few years, we are finally starting to see the fruits of all that labor, with a number of gene therapies being approved for serious genetic disorders, including diseases like spinal muscular atrophy, an inherited form of blindness, and the gene therapy for hemophilia B that I just mentioned. For Huntington disease, the gene therapy being tested is called gene regulation. It is designed to reduce the production of a protein called huntingtin, the buildup of which is thought to cause the symptoms of Huntington disease. Our investigational therapy is called AMT-130. You might wonder, why AMT? Well, our company was originally called Amsterdam Molecular Therapeutics. All of Unique's development programs have used the term AMT prefix back to the early days of the company. So there you have it. AMT-130 blocks the messenger RNA, which plays a role in making the Huntingtin protein, thereby preventing its production. Next slide. So how do we get AMT-130 where it needs to go? You will see on these panels on the slide. First, during a single surgery, AMT-130 is administered under general anesthesia by a skilled neurosurgeon using real-time MRI guidance into specific areas of the brain which are most affected in HD. These are the caudate nucleus and the putamen, together known as the striatum. Second, AMT-130 delivers instructions to make blocking messenger RNA that prevents the production of the Huntingtin protein. And 3, AMT-130 is designed to stay in the brain cells and continue to make the blocking messenger RNA, preventing the production of the Huntingtin protein over time. Next slide. So what are the goals of gene therapy for Huntington disease? By definition, goals are aspirational. Ours are to prevent or at least delay the progression of Huntington disease symptoms, establish long-term benefit, and maintain the quality of life of those with HD. And most importantly, we have to show that we can do this safely and effectively in our clinical trials. Next. Now let's take a look at the currently progressing clinical trials evaluating AMT-130. There are 2 such trials called HD-GeneTRX1, which is in the US, and GeneTRX2, which is in Europe. In both trials, there are a low dose and a high dose group. We started with a low dose because these are early trials, and primarily their focus was to assess safety. Once the safety of the lower dose had been confirmed by independent experts, the trial moved to the higher dose group. Some patients in the GeneTRX-1 trial in the US went through an imitation surgery in order to compare them to people who got the study drug. In addition, the trial was blinded, meaning that neither the patient nor their doctor or ourselves, the company, knew who got the study drug or the imitation surgery. After 1 year, the patients were unblinded and knew if they got the study drug or not. And those who didn't could then decide if they wanted to cross over and receive the study drug if they're still eligible. Patients that were able to cross over have now done so. I'm also happy to report that a third group, or cohort 3, as you can see on the slide, is now moving forward in both the US and in Europe, in both the low and high dose groups. This third cohort will not have the imitation surgery, and patients will be split evenly between the low dose and the high dose. Next slide. This is a bit of a complicated slide, but I'm trying to walk you through it, and I'm not going to go into a lot of the details. But this gives you a sense of the trial design and the eligibility criteria for the trial. These determine who is eligible to be considered for the trial. However, even if you meet all these criteria on paper, you still need to be assessed by the site to enter the trial. As I said, I won't go into the details, but I will highlight some of the major requirements. First, we need a clinical diagnosis of an early manifest HD. Second, a genotype of 40 CAG repeats or greater. You must be between 25 and 65 years old. However, people with previous exposure to gene therapy, to Huntington lowering strategies, or experimental brain surgery would unfortunately not qualify. One important criterion is striatal volume. I mentioned before that the striatum is the part of the brain that is most affected by HD. That area needs to be of an adequate size to safely receive the gene therapy. Now, if someone is interested in participating, they'd first need to do a screening visit, as you see on this slide, to see if they qualify. And if they do, they would then have to do what we call a baseline visit, which is serving to gather data and information to measure against later. They would then undergo the surgery, and then we will be following these patients on a regular basis every 3 months in general for the first 2 years, and then every 6 months between years 3 and 5. During those visits, the various safety and efficacy measures that you can see on the right-hand side would be assessed to see the impact of AMT-130. Next slide. One more, please. So over the next few slides, I will be describing the results from our latest update, which included the information we had as of the end of September 2023. When we reviewed the safety results, we concluded that AMT-130 was generally safe and well tolerated in both the low and high dose. We did observe a number of serious adverse events that were related to AMT-130, particularly at the high dose. However, these were believed to be due to a swelling of the brain, which responded well to a short course of corticosteroid therapy. That is why in the third group that is currently progressing, we included a short course of steroid therapy to prevent or minimize these potential adverse events. Next slide. In the next few slides, I will show you the efficacy data. These consist of measures of functions, motor symptoms, and cognition. Finally, I will show you a composite score called the Composite Unified Huntington Disease Rating Scale, a mouthful. We call it cUHDRS for short, which combines all of these measures into a single score. Next slide. I will be going through a series of slides here, which will depict the data in the same manner. So I'm going to spend a little bit more time on this slide to kind of help situate things. The green line on these charts will depict the patients who received the imitation surgery and who were followed only for 1 year. That's why you see it stops at 12 months. Because the effects of AMT-130 are expected to last much longer, we needed to find out how our patients would have progressed had they not received the drug. So we turned to the CHDI Foundation and searched through their database to identify patients with similar characteristics to those in our trials. Their information is depicted by the orange dashed line on this and the subsequent charts. The scale we are showing on this slide is the total functional capacity. It consists of 5 items measuring the ability to function at work, manage finances, perform domestic chores, activities of daily living, and care level. As is shown by the orange dashed line, patients are expected to lose about 1 point every 2 years on this scale if left untreated. In this graph, down reflects worsening of symptoms. As you can see, AMT-130 treated patients appear to deviate from this course. They seem to maintain the same level of functioning on average, particularly starting the second year after surgery and on. We're very excited by these promising results, but we would caution you that we are still in the early days, and as you can see from the numbers in the table below, we have fewer patients who have been treated for longer than 18 months. We're continuing to follow these patients, and we look forward to provide further updates on their progress in the future. You will also notice that the patients who got the imitation surgery and were followed for 1 year, as depicted by the green line, did not progress much. We believe this is because 1 year is too short for these changes to be clearly seen, particularly in early manifest patients, and possibly also because of what we call a placebo response, which I'm sure you've heard before and you're quite familiar with. Next slide. Here we show the results from the total motor score, which is a scale that measures things like muscle rigidity, tremor or shakiness, chorea or jerky movements, and balance. In this case, a lower score is better. Here also we see that both the low dose and the high dose groups appear to remain on average around baseline, suggesting preservation of function, and clearly performing better than the orange line. We also see that the high dose depicted in the blue line seems to be doing a bit better than the low dose depicted in the purple. Next slide. This and the next slide will depict 2 cognitive tests. Familiar with it. Lower scores here indicate impairment. So here too, both the low and high dose seem to perform close to their baseline and better than the orange line. The last 2 measurements, however, in the low dose, as you can see in the purple line, 1 patient had a lot of variability between visits, pulling the purple line up at month 24 and down at month 30. So let's look at the next slide with the simple symbol-digit modalities test, which is another measure of attention and processing. In this test, we find the strongest evidence of separation between the low and high dose group, the low dose being the purple and the high dose being the blue, teal blue. While we do not know for sure why we see that, it is possible that this test relies more heavily on parts of the brain, like the cortex, where more of AMT-130 is transported when higher doses are administered in the striatum. Next slide. As I mentioned before, this is the composite score, or cUHDRS, which is a sensitive measure for worsening of symptoms, particularly in early manifest patients. In fact, it has been shown to do so in a recent large study in HD called Generation HD, which I think you are quite familiar with. Here you can see that both doses of AMT-130 appear to perform better than what would have been expected if they did not receive any treatment, as shown in the orange line. We also see that the high dose, again depicted in the blue teal, do better than the low dose in purple, and seems to maintain the same score as baseline, and perhaps show some improvement even by month 18. I draw your caution that month 24 only included 2 patients at that high dose and therefore should be viewed with a lot of caution. Again, I have to also caution you that this is an ongoing study, and we will continue to report as we go along, and there will be more patients that are going to be We will have more data with longer-term exposure of these patients going forward. But we're very happy with what we've seen so far, and so far, so good. Next slide. So overall, as of this last update, we feel positive about the clinical efficacy we're seeing. Both doses seem to perform better than the natural history group, particularly as the observation period extends beyond the first year. We're also encouraged by the initial evidence of a difference between the doses, with the higher dose showing better efficacy. This suggests that AMT is doing something to stabilize the disease, and giving more of it seems to lead to better results. Next slide. I have a few more slides to describe the biomarker and imaging results. So next slide. Here we're showing the results of a protein called the neurofilament light chain in the cerebrospinal fluid. This is known as NFL for short, and it has nothing to do with football though. NFL is a sensitive marker of injury to brain cells. So maybe it has something to do with football, but we're not going to talk about it today. That explains why you see an increase following the surgery, as you can see by both the blue, the teal blue, and the purple lines going up at month 1 and then coming back down. We believe that the peak increase is related to the surgery itself and not to AMT-130, because we do not see a difference in the magnitude of the increase between the low and the high dose. If that was the case, we would have expected to see a difference, with the high dose showing a larger peak. And we see the levels returning towards baseline by 1 year for the low dose for sure. Now, I draw your attention at month 24, and it's hard to see this, but trust me, there's an orange dot at month 24, that represents data from natural history cohort, which Dr. Wild actually, which is a study that Dr. Wild conducted, and he perhaps can say a few words about it at the end of my part of the presentation today. But as you can see, both doses are lower than that, what would be expected at that time point in our study. In the next slide, I'm trying to show you the fact that we had one patient in the high dose, which is still depicted in the blue teal, experienced an immune reaction, that patient, and some swelling of their brain, which responded to initial course of steroids. That patient had higher NFL levels compared to the others and were an outlier. And so what we did is we showed the data that if we remove this patient, as you see in the darker blue line, you see how the levels drop down and they go close to baseline by month 15 and actually below baseline at month 18. Again, like I said, month 24 could be disregarded because it's 2 patients and it's very difficult to draw conclusions from only 2. We will be in better shape in discussing this in the next iteration when we show the data, when we have more patients who reach that time point. Next slide. Here we show the levels of the mutant Huntington protein in the cerebrospinal fluid collected from the lower back. The concentration of MHTT are extremely low, making the assay to measure them very difficult to perform. We believe that measuring very low levels of MHTT in the lower part of the back is not a good way to evaluate the effects of AMT-130, which is administered in the striatum, a relatively small brain structure buried deeply inside the brain. Next slide. Lastly, here we show the total brain volume we measured by MRI, or magnetic resonance imaging. Here we see that both doses seem to be below the natural history. We believe this is indicative of the brain reaction to the surgery and not evidence of acceleration of the disease, which would have been manifested by worsening of clinical symptoms and an increase in NFL over time. On the contrary, as I've shown you before, we see stabilization of the clinical symptoms and lower level of NFL over time at both of our doses. Next slide. And one more, please. So in conclusion, the latest clinical result update is exciting, as we believe AMT-130 is showing early promising signs. We believe that AMT-130 is generally well tolerated with a manageable safety profile. There are promising efficacy signals suggesting preservation of function. This is more apparent with the high dose, suggesting a dose-response relationship. The NFL data, a sensitive marker for neuronal injury, appear to be supportive as well of the effects of AMT-130. So we believe the totality of these data are encouraging and they support continued development of AMT-130. Next. So let's talk about the next steps. One more, please. What we have done is we started enrolling patients in the third cohort, as I described earlier, to better understand how to manage the immune reaction and improve safety, tolerability, and possibly efficacy. We plan to discuss the results I just showed you with the regulatory authorities, both in the US and in Europe, to agree on a path forward for getting AMT-130 approved and available for patients. Lastly, we plan to provide another update of these results by the middle of this year. Next slide. For more information, most importantly, talk to your doctor, talk to your nurse, talk to members of your care team. That is always the best place to start. Since you're here, you already know that patients' organizations like the HDSA offer amazing resources and education. If you'd like to learn more about where the clinical trial sites are located, please go to HDSA's hdtrialfinder.org that Leora just spoke about earlier today. You can also go to clinicaltrials.gov, and you can always feel free to email us at amt130_clinical_trials@uniqure.com. And with that, let me please turn to Professor Ed Wild. Ed?
Hi, everyone. Thanks, Walid. Shall we stop showing slides? Because I don't have slides. I just have my face. Sorry about that, if that's a big disappointment. May I start by saying to all my US friends, Happy Pancake Day. If you don't know what that is, you'll have to Google it afterwards. Quick disclosure, interest statement on my part. As Walid says, I'm an investigator on this trial. I'm also an advisor to Uniqure, but I'm also an investigator and advisor on multiple drug trials in Huntington's disease and therapeutic programs, including some which lower Huntington and some which have a different approach. Essentially, I'm happy to help anyone who's trying sincerely to develop effective treatments for HD and is interested in hearing my opinion. My personal academic background, as Leora said, is in biomarkers and clinical trials, but I also spent a long time drawing around brains on MRI scans during my early days, so I know a bit about the MRI side of things too. I'm going to be quick because there's amazing questions coming through and we need to leave lots of time to focus on those. So I'm just going to give you some sort of high-level thoughts that Uniqure have not attempted to control. I think the really important thing here, and I'm seeing lots of excitement in the Q&A, this is a very exciting program and these results are exciting, but we need to keep everything in context and bear in mind the broader picture, which is that we don't have anything that we know works to slow the progression of Huntington's disease. And this is the first ever gene therapy trial in Huntington's disease. And as we've heard, it involves major brain operation to inject this gene therapy drug into the brain. And the hope is that that will produce lifelong or extremely long-lasting effects, reducing the concentration of Huntington protein, and that we have the right to expect that will produce improvements or at the very least slowing of progression of Huntington's disease. However, it's a trade-off, and we know that going in. This is brain surgery is no small thing for anybody. For someone with Huntington's disease whose brain may be more fragile than someone who doesn't have that genetic issue, brain surgery may be an even bigger deal. So, and the fact that this is a treatment that's long-lasting has an upside. If it works, it has a potential downside if there are issues, because there's no off switch for this treatment. So we know going into this program that it's a sort of potential high-risk, high-reward situation compared with something else that's a pill that you take, and if you have problems, you stop taking the pill and then the thing is no longer in your body. So, and thankfully we have a lot of drug companies engaged in Huntington's disease now. And many of you may be kind of used to hearing these updates that sound good. what I think we need to bear in mind is that this program is fundamentally different from any other program that's currently running in HD. Pill trials we've been doing for 20 years or more. The injection of drugs into the spinal fluid was new in 2015, but we've heard multiple updates from that program. This is the only gene therapy program in Huntington's disease. And at all times, although we're hearing some quite fine detail about the outcomes, we need to bear in mind that fundamentally this is a first in human program of a new gene therapy, and essentially we are discovering new science. And in a program like this, the company proceeds cautiously, which is appropriate. We enroll patients quite gradually, and we follow them for a long period of time, because in a program like this, benefits as well as potential issues can emerge quite slowly. Essentially, this is a road that we need to pave while we are walking it. At the very basic level, the early years of this program have involved figuring out how to do brain surgery on patients with Huntington's disease and how to inject this drug safely into the parts of the brain that we want to treat. And one of the issues that Walid mentioned that's come up as a discovery that we didn't necessarily expect it, but when we saw it, it wasn't a big surprise, was that in the few weeks after surgery, there's some inflammation in some of the patients in the brain. That's unusual to see in Huntington's disease, but it's not unusual to see in patients who've had brain surgery or patients with other neurological issues. So as neurologists, we have a good idea how to manage that, and that's the corticosteroid treatment, drugs like dexamethasone that Walid mentioned. So again, this is a discovery, and we finesse how to manage these issues as we go through the program. So safety first and foremost, and that looks really good. And that's why Walid was able to focus on the outcomes for so much of his presentation, because the safety so far looks good. So what about the outcomes? Now, there's a lot of measurements taken. We've seen the clinical measures, we've seen cognitive, motor, functional, imaging, biomarkers. It's a lot. One thing matters more than anything else, and that's how the patients are doing. That's the clinical measures, okay? So many of you will be familiar with things like the total motor score or the total functional capacity. Those are components of the standardized neuro examination and questionnaire that we've been doing for decades in Huntington's disease. The composite UHDRS or CUHDRS is more recent and less familiar, but the ingredients of that score are familiar. So the composite UHDRS was designed to be sensitive to change in Huntington's disease, whether that is deterioration or hopefully sometime improvement. And the way it works, it's a composite. So it basically takes different components that we're familiar with, like the motor score, the functional score, and some of the cognitive scores, and it squishes them into one number, and that's why it's a composite. The fact that it's sensitive to change, we know already from programs like the tominersen program, in which, unfortunately, we saw the composite UHDRS score declining, which is a sign that those patients were actually worsening more rapidly than expected. And what we want to see is that score going up as time goes on. So, if we're not, some drugs might be better for movements, some drugs might work better for cognition, some might work on function alone. If we don't know exactly how a drug is going to produce harm or benefit, the composite's a very useful thing to look at because it's capable of capturing change from across the disease. And I don't have slides, but I am going to hold up the slide that I think is important from Walid's talk, which is this one, the CUHDRS. So that is the composite measure that he showed you. And the one that's the blue line that seems to be going up is the patients treated with high dose of the AMT-130 gene therapy. And, these are small numbers. The numbers below the graph are as important as the lines in the graph, but that line is heading where we want it to head. It is not evidence that the drug is working, but the line is where we want it to be right now in order to proceed with the program and see whether that line can stay where it needs to be. Oh, thank you, Dan. He shared it. Sorry, I didn't prepare anyone for that possibility. I've actually finished talking about that graph now. So you can stop sharing again. We can look at the ingredients of the composite if we want to, but they're all important in their own way. But the total motor score, I think, is one that would change rapidly if patients were worsening, and the total functional capacity is the one that we know declines most steadily and regularly as the Huntington's disease progresses. So it's good to see those seeming to move towards the direction we want. Again, this isn't proof that it works, but it is comforting that the graphs are on the side of the good or bad line that we would like right now. So what about biomarkers? I'm a biomarker person, and they tend to produce confusion. I'll start with mutant Huntington, which we measure in the spinal fluid of patients, but we measure it by sticking a needle in the spine right above the what English people call the bum, the very base of the spine. So it's about 4 feet away, 3 feet away from your brain where the injection actually goes in. It's not necessarily something that we can expect to see lowering of mutant Huntington in that fluid from down at the bottom of the spine when a drug is being injected into a small volume of the brain at the top. I would see decreases in mutant Huntington as being really nice if we see them in this program. But as I've said before, and you can look back through old HDSA presentations I've made, it's entirely possible that this drug would meaningfully lower Huntington in the brain right where we want it to, but without actually being able to measure those reductions in the spine. And there are technical reasons as well why that might be difficult, because there's quite a bit of variability if we measure the same drop of fluid twice, we sometimes get slightly different answers from that measurement system. And we're right at the limit of being able to detect it in these patients who are relatively early in the disease. The other major biomarker that's been presented is neurofilament, or NFL, not the football thing, but the protein that is released from damaged brain cells. And there's complexities to everything, but so far in Huntington's disease, NFL seems to be behaving quite straightforwardly. It is increased in people with the gene, the HD mutation. It gradually increases more as time goes on and as the disease symptoms start to worsen in the brain. And things that make HD worse tend to increase NFL. And if we see NFL going down, most people like me would interpret that as a good thing. Now, whenever brains are operated on, you expect to see a big release of neurofilament, of NFL, and so it's not surprising that we see that NFL rising. What we then want to see is it gradually returning to baseline and hopefully ultimately either below baseline or below where it would otherwise have been. And as Walid mentioned, in my study, the HDCSF study, we did see NFL slowly increasing as time went on in Huntington's disease. And so even if we are merely flattening NFL at the moment, that may well be a better thing than if NFL was continuing to increase. But again, although NFL is pretty reliable, we can measure NFL much more accurately than we can measure mutant Huntington. It's not the be-all and end-all, and the thing that matters most is the clinical changes. I won't go on about brain imaging. I'm happy to answer questions about that if they come up. What I would say is that this is first and foremost, the patients who volunteered for this program are amazing. They are, I said at the time when I learned about the first patient being treated, they are the Yuri Gagarins of Huntington's disease. They have volunteered to undergo something pretty major as a service to mankind, to humanity, and that's a remarkable thing. The company behind it also needs to be commended, I think, for being so committed to a program which does have a degree of risk involved and the potential for huge reward if, and by which I mean patients getting better if the drug is working. We need to take it slow and steady. We pave the road as we walk it. We learn as we go along. Safety is more important than anything. Biomarkers are useful, but the clinical outcomes are most important, and time will tell where this program is headed. I've spoken for long enough. Let's hand back to Lior, I guess, for questions.
Thanks very much, Ed, and thank you, Walid. Thanks for that very accessible explanation of the latest data. We've got tons of questions coming in. We've got more than 300 people here, so we're going to do our best to answer as many as we can today. I want to set expectations, especially for folks who are coming from industry positions, that we're really going to focus on community questions today. And what I would like to start with is kind of a practical question. So people often have these very practical questions about participating in any kind of HD research study. So maybe you could talk a little bit more about what it's like to be in this trial, what happens the day of the surgery, what the follow-up is like. I'm not sure, maybe that's a question for Walid.
I don't know. I wanted to see if I'd wanted to speak about it because you are an investigator, but I'm happy to talk about it, Ed. So let me know.
You go first, and then I'll chip in from an investigator point of view.
Right. So as I said, that the first part is actually doing the screening to make sure that you meet the criteria for this, because at the end of the day, as Ed said, the most important element is to make sure that we do this in a safe way. So that's why we need to make sure that we have the right people for for the study. We do a number of blood tests. We do some imaging to make sure that the size of the caudate and putamen are of a large enough size that we can safely inject 130 there. There's a lot of work that happens with a neurosurgical committee to decide on the trajectory. All of these are done in preparation of the surgery. And if all of this is positive, the surgery is scheduled. The surgery is a long surgery because it has to be done very, very carefully. There's a frame that is set out in the brain. This is not highly unusual. There's many approaches that uses these frames, from treatment for epilepsy, or even deep brain stimulation for Parkinson's. So it's not really something that has been uncharted territory. But it's a complicated procedure that skilled neurosurgeons who have experience with this will get to do it. The surgery lasts anywhere between 12 to 14, 16 hours. I think over time, we're aiming to make this shorter, and that's one of the aim of the things that we want to learn, how to make this shorter without taking any shortcuts, or actually having to make sure that safety is maintained. And then often people stay in the hospital after that for anywhere between 1 to 3 days, and they're actually surprisingly get released after that. They're doing okay. They get monitored initially very closely. We have either phone visits or in person shortly after the procedure, at 1 week, 2 weeks, and then at 1 month, and then at 3 months. And if need be, in between, we bring people in. We often do not need to. We've had people in our trial have gone for about 30 plus months right now, and towards the later parts, just people come in every 6 months and get some blood tests, the clinical assessment, some imaging as well, and off they go. The beauty of this treatment is that you give it once and then it stays there, and hopefully it continues to work. That's what we expect to see. The clinical data that we're seeing so far suggests that it's doing so. And as Ed said, the more we wait, the more people will progress. I think one of the questions that I've seen in the chat, and maybe I can address now, why are the numbers reduced over time? Well, it's not that people have been leaving the study. It's people have not had the time yet to reach to that time point. So we keep on monitoring people, as I said, for 5 years, and the reason why you see those numbers lower at the outer time points is because simply people haven't had the chance yet to make it to that time.
Maybe I'll just chip in from the investigator side. From there's a couple of slightly surprising things. One I think it's been a surprisingly difficult trial to get into. because there's been a lot of enthusiasm, but the inclusion criteria for this trial, and you probably know inclusion and exclusion criteria for trials are always pretty strict. For this trial, it's even harder to get into because having gone through all of the clinical assessments, unusually, we then need to do a brain scan and make sure that the volume of the brain that's going to be injected is actually big enough to start with, because if there isn't enough of it there, the surgery is not safe. And so that, I think, has been an extra level of complexity. And so if you're potentially thinking about being screened for this trial, one of the things to bear in mind is it's slightly more likely in this program that you might find, for one reason or another, including the MRI criteria, that you're not able to actually go ahead and have the procedure. The operation itself, as Walid says, it's a major operation. It's 10 or more hours of surgery. From the patient point of view, they're asleep, so they go to they have the anesthetic. They wake up, they've got a sore head, but broadly speaking, recovery from that procedure is pretty quick. And from the surgical point of view, the surgeons find it quite boring. I have to say, no pun intended, because they have to make holes in the skull and then find the right spot. But a lot of it's done with computer assistance, and then the bulk of that time is actually very slowly injecting the gene therapy drug, which takes several hours. However, they pay close attention throughout. And then that flurry of excitement, few days in hospital, is then followed by the patient essentially returning to normal life and undergoing these initially quite frequent and then less frequent follow-ups. So it's an unusual time. Lots of assessments before, a flurry of excitement during the surgery and afterwards, and then back to normal life for 5 years, and hope that the drug will start to show its beneficial effects.
Guys, I want to say thank you both for the presentations you gave, and to echo something that Ed said earlier, I know there's people that are on this call that are participating in the study, and want to thank you all for being a part of this. And to folks that have participated in other studies too. As Ed indicated, if you were not doing this, science would not be moving forward, and we would not be talking about things like we are today. So huge thank you to all of you for that. There's a couple of questions that have come up that are a little bit more forward looking, and I hope you guys might be able to touch on this. We're all very hopeful for the results of this trial, especially as it goes into later phases, and we have the potential to be going towards clinic. How are you already, or are you already talking about how are you working on things like access to medication? How are you working with things like cost and how this might work with insurers? I think people know that, the science and the clinical results that we're looking at are really great, but if this goes to clinic, what already is being done? What's being done to prepare for that?
Yeah, so maybe I can address that. So we do have that on our radar screen, but honestly, I think it's way too early for us to get down that path. I mean, as Ed said, this is the first study we're doing. We are moving, and I know sometimes we're like impatient. We would love to have more data and include more people, but again, we needed to do this carefully. It's a major surgery. It has not been done before. We're pioneers. We're blazing new path, but we cannot be cowboys about it. We just need to take this very seriously. There's people's lives at stake here, and if we make a mistake, we could ruin potentially good treatment for a lot of other people. So going slower so that we can arrive to our destination where we're supposed to go correctly would be the good way to go. Even if that means that we should not get this drug to market because this drug is not good enough, going slower, finding that out is the best way to go. All of this is to say that it is on our radar screen, but I can guarantee you we have not been discussing pricing or anything like that, but we're definitely very keen on access, because what good is it to develop a drug, get it regulatory approved if the patients cannot get it? That's not what we're here for. So it's all part of this. We will be working towards that. We're very keen on getting this to the patients, but I cannot share with you any more details right now because we simply don't have them.
Thanks, Walid. I think we've got some questions that are sort of in a similar vein around access, but really the questions are about who this drug could potentially help. So, these early safety trials are focused on a very specific population of people with HD. The eligibility criteria are very narrow, but of course, there are many people who are wondering whether there's the potential to delay symptoms or to slow symptoms in a much later stage of HD. So maybe you could speak a bit to Unicure's plans for investigating AMT-130 in other populations with HD, or just in general, the idea of when a drug has been tested in a particular population, whether that could potentially become available to others.
Yeah, no, that's a very good question. And it's always like that in drug development. You start from a narrow, patient base to maximize the chances of standardization, the size of the brain, the certain level of symptoms that we have, so that we can match those to a correct natural history that we can trace. We try to target earlier parts of the disease because if this is meant to prevent further decline, you want to try and intervene early to preserve the most function. But once you establish this, and I saw questions about age and stuff like that, then you start loosening these kind of criteria, because then you want to make this more available. But again, you just have to do it one step at a time. You need to establish that indeed in patients of maybe a younger age, patients who have more advanced disease could still benefit, and those will come in subsequent trials where you can start to enlarge the patient population and monitor this. I think the burden to show the level of efficacy that you would do in say, what we call a pivotal trial, the one that would serve as the basis for registration and getting approval from regulatory agencies, will become a bit less as you are broadening, because then your focus is to make sure that the data are coming in line with what you would be expecting from safety, but also from efficacy. But yes, definitely our objective is to broaden as much as possible, not only to those who are a bit more advanced, but also to those who are even in pre-manifest period, because you want to try and stop things from happening. If this is actually drug is showing, this therapeutic intervention is showing that indeed blocking the formation of this Huntington protein is indeed stopping the progression of the disease, or hopefully stopping, but maybe slowing it down significantly enough, then you want to intervene as early as possible, but also later. There's no reason why we shouldn't do that. The only thing is later, if there's a limit and the size of the striatum, we might need to apply different techniques. But again, those are also part of our thinking, because as Ed said, one of the difficulties right now is to be able to get people with the right size of striatum, caudate, and putamen, so that we can approach them safely. But as we are perfecting this technique, we can also try to develop better neurosurgical approaches that potentially could allow us to lift some of those limitations and broaden the patient population. But again, one step at a time.
Sorry for dropping off there real quick. Magic of technology, of course. There's a couple of questions that have come in about imaging, like how is the brain being measured? And since it's tough to measure Huntington, what other measures are most important?
So Ed, I'm going to punt this one to you, since this has been your expertise.
Yeah, so the imaging that's used in this program is MRI scans, which are the kind of thing that have been used in many other trials in Huntington's disease, and have also been used for many years now, decades, to study the natural history of the disease. And what's focused on is a type of MRI that it can quite accurately measure the volume of the whole brain or of individual regions of the brain, such as the caudate and putamen. And we know that the whole brain, well, if you're over 25, your whole brain is shrinking while I'm talking, even if you don't have Huntington's disease. So everyone's brain is shrinking the whole time. In Huntington's disease, this shrinkage occurs more rapidly because of the damage that the gene is doing. particularly in the striatum, but in when we measure the cortex, which is the crinkly bit on the surface of the brain as well. That's the main type of imaging that's being used. Unfortunately, we don't have the kind of scans that are sometimes used in other diseases like Alzheimer's disease that can actually look at the level of individual proteins. So in Alzheimer's disease, you can do something called a PET scan, positron emission tomography scan, and it basically lights up parts of the brain that have amyloid or tau or proteins of interest, thanks to a radioactive injection that sticks to those proteins. We don't have that yet in Huntington's disease, but it's something that CHDI Foundation is leading on and has been making excellent progress with. That would be really useful in this program, because what we'd be particularly interested in is looking at those deep brain structures that have been injected and seeing whether the level of Huntington protein has been reduced. We don't have them yet, but the good news is that if and when those scans become available, even if this program isn't being actively pursued anymore, which I hope it will be, the patients who've been treated may still show reduction of Huntington because of the fact that it's a one-off treatment. So hopefully those two technologies will come together and we'll be able to look inside the brains of these patients and see what's happening. What are the volumes telling us? I think it's really difficult to know, because the volume measurement techniques were developed in healthy brains and in the brains of people whose brains are shrinking. What's happening here is something very different, which is that holes are being drilled in the head. I'm sorry to be graphic about this. And then tubes are being pushed into the brain and something's being injected. And then in many patients, inflammation is occurring. And the MRI scan's good at picking that up, but it can, because that can cause swelling, it can kind of mess with some of the volume measurements. In other words, if the volume of something changes, we won't necessarily know whether it's because there's been an effect on the disease, or whether it's an artifact of the surgery, or whether it's an artifact of inflammation. So when I think about the brain imaging results in this trial, I think of them primarily as a safety measure, because some of the scans are really useful for picking up the kind of inflammation and the kind of swelling that we might really need to know about to look after the patient safely. What they're less useful for, I think, is the sort of measurement of slow change over time in volume that we might want if we're going to use MR imaging as a biomarker. Thankfully, we have multiple biomarkers that we can look at, neurofilament being one, and the clinical measures, as I've said, are more important than whatever else is going on. But when we look at a program like this, it's great to be able to look at the same thing from different angles. And if lots of things are pointing in one direction and one thing is pointing in another direction, we don't disregard that thing, but we need to ask ourselves how much we understand about what it's telling us, and what are the results overall sort of pointing towards when we think about them all together.
Thanks very much, Ed. I want to respect everyone's time, so I'm going to ask one more question, and then we'll wrap up. And certainly we can help with any follow-up if folks want to reach out to HDSA. So we're seeing some very early but promising data here in very small numbers of people. This is a treatment that lowers both normal Huntington and expanded Huntington. So can you speak to whether you think the clinical data is any reflection of the safety of that normal Huntington lowering? I know this is a big question for folks in the field.
So maybe I'll tackle this first, and then I see if Ed, you want to add a few things. So when we were choosing the doses to use in our trial, we based this on our preclinical models, and we were keen not to reach a level of inhibition of both the wild and the mutant Huntington protein to like 0. So we dropped by I think 75% was our target. I think looking at our clinical data, and again, that's the best measure that we have right now, we do not see any evidence that actually higher dose does worse than the lower dose, which suggests that we're not getting to the point where we're reducing the wild type to a level that is now starting to backfire, so to speak. So that's really the best I can answer at this point. Again, the caveat being that we are, again, small numbers, and we are looking at those over time. But So far, the evidence is starting to show that actually the high dose seems to be better than the low dose. And as if this continues to be the case, as we keep on looking at the data in the next 6 and 12 months, I think we will, those will be the data supporting such a statement. Ed, do you want to add anything to this?
Yeah, I'm probably a bit philosophical about this. I think it's easy to imagine a perfect treatment for Huntington's disease, which is one that takes the mutation and turns it into a healthy gene, or takes the abnormal protein and turns it into a healthy protein. We don't have those treatments, and any hope of giving them to our patients is pretty far in the future. So we mustn't let the perfect be the enemy of the good. And so we need to figure out what we can accomplish now and in the near future. There are treatments being tested that only lower the mutant protein, while or selectively lower the mutant protein, but those are much more difficult to design, and they don't necessarily work for that big of a percentage of the whole HD population that we would like to treat. So, this sort of approach has innate advantages, namely that it's a single treatment that lasts a long time and could be given early to produce a lifetime of benefit. The one thing we know for sure is that doing nothing results in bad outcomes in Huntington's disease. So we don't want we do want a perfect treatment, but we can't throw away treatments just because they might not be perfect. Nor do we need to think of a potential safety concern from lowering wild type as a deal breaker. We don't need treatments to have no side effects. We need treatments that are better than having Huntington's disease. So if there is a potential downside from lowering the production of the normal Huntington protein, and we don't know for sure that there is, or rather we don't know in humans what percentage is tolerable, if there is a potential downside to that, it really only matters a great deal if that downside is worse than doing nothing. In other words, lowering the mutant protein might be great. Reducing the wild type might take the edge off that benefit, but if we're producing benefit overall, it's still something that we want to do. I didn't tie myself in knots too much there. I guess I'm trying to say we need to keep an open mind, figure out what's causing what as we go along, and start building from where we are now, no treatments, to something that works, and then get a foot in the door and take it from there.
Ed, Walid, again, we just want to thank you for this presentation and sharing what you are learning and what we as a larger community are learning in this process. And I think, Ed, you said something that I think we all need to just remember is, while treatments are being developed, the goal is to change the way that people are living with Huntington's disease. And we only learn that as we're doing these trials. I just wanted to say that doesn't mean there aren't things we can be doing right now, because there are. And I think, there's a couple of things that came up in the question. I would just like to point people to the HDSA.org website. If you are looking for support, if you're a member of a trial, if you are a member of an HD family, if you are looking for resources, please know that there are things out there right now. And HDSA.org has a wealth of resources, from community services to advocacy opportunities. And just say, while the work is being done, like the team is doing at Uniqure, there is work being done to help people live better with this disease today. So tune into that. Leora, want to go ahead and wrap this up?
Yes, I just want to thank our speakers, Walid and Ed. Thank you so much for making this accessible and for answering community questions. And thank you to the more than 300 people who came to attend this webinar today. I truly believe that, staying informed is a form of advocacy. So thank you. I hope that you'll find support from us and join us in our advocacy efforts as well. And I hope you'll join us for our next HDSA research webinar, and we'll be in touch about that. Thanks, everyone.
Thanks a lot. Bye-bye.